Did you take down the heading? Experiments related to which one? Experiments related to chemical basis of life. Okay. Did you write the heading? Rima?
Yes, sir.
Right. So, first of all, we are going to work for test for home. Test for home. Test for
Reducing sugars.
reducing sugars. Who are reducing sugars? First thing, any sugar that reduces Benedict's solution and produces a brick red color precipitate is home. Home.
Reducing sugars.
Reducing sugar. Okay, when you heat it with Benedict solution, a brick red color precipitate should be made. If that precipitate is coming, it is a reducing sugar. In exam paper, if they ask you the definition for reducing sugar, any sugar that reduces Benedict solution, okay, for any sugar, you can write this way also, any sugar that produces a brick red color precipitate, okay, when heated with Benedict's solution is called reducing sugar. Here, all monosaccharides are what kind of sugars? Reducing sugars. Any monosaccharide, glucose, fructose, galactose, ribose, deoxyribose, okay, whatever the monosaccharide coming, all the monosaccharides are coming under reducing
Sugars.
Secondly, all disaccharides except sucrose. Sucrose is not a reducing sugar. Are you clear?
Yes, sir.
Good. Please mark it down. Completed.
Yes, sir.
Right. Now, let's have a look on this video. Have a look.
We are going to perform the Benedict test. The apparatus used for this test are test tube stand, a test tube holder, measuring cylinder, and a beaker containing the sample. The reagent used for this test is Benedict's reagent. Take a test tube and hold it firmly with a test tube holder. Add 5 ml of Benedict's reagent. Add 8 drops of the sample. Shake the contents well and boil it for approximately 1 minute. Shake the test tube by heating it. There is a formation of a color change which indicates the positive Benedict test. We are going to perform the heat coagulation test. Take a test tube and fill the 2/3 part with the urine sample and hold the test tube firmly with the test tube holder. Heat the upper 1/3 part.
Let's go on to the next one. Right. So have you seen the practical? Have you seen the practical?
Yeah, sir.
Yeah. So what is the color of Benedict solution?
Blue color.
Blue color. When we heat it with
Test tube holder.
Benedict solution, the glucose solution,
Add 5 ml
you
of
are
Benedict's reagent.
getting a brick red color precipitate, right? Brick red color.
For approximately 1 minute. Shake the test tube by heating it. There is a formation of a color change which indicates the positive Benedict test.
Right. So this is what we want to discuss here. Right. So what is the procedure? What is it? Procedure. What does your syllabus tells how the way to do this? Okay, the way to do this, the method to do this. That's the procedure. This is the procedure. This is the procedure. What is the procedure? You are taking 2 cubic centimeter of a solution of a reducing sugar. You can take a glucose solution, you can take fructose solution, galactose solution, lactose solution, maltose solution, anything is okay. But normally available one is home, easy material is home.
Glucose solution.
Glucose solution. Add equal volume. That means you are taking 2 cubic centimeter reducing sugar means how many cubic centimeter Benedict solution you will be adding?
2 cubic centimeter.
2 cubic centimeter. You have to well shake it and you have to well boil it. When you do it, what you get? What is your observation? A brick red color precipitate. Okay, would be obtained. Clear?
Yes, sir.
Yes. So could you please complete the test for reducing sugar? Completed? Have you completed? Good. So, secondly, I am going to speak about test 4 for non-reducing sugars. Okay, fortunately, non-reducing sugar means it is one and only sucrose there. Which one is the non-reducing sugar? Which one is the non-reducing sugar? Right. So, it will not give a brick red color precipitate when heated with Benedict's solution. That is confirmed. That kind of sugar only we call, what is it?
Non-reducing sugar.
Non-reducing sugar. Now you are heating a sugar. It is not coming. You take some sucrose and heat it with Benedict solution. Will you receive any precipitate? You take some sucrose solution. Okay. Proceed here. Take some. Okay. Take 2 cubic centimeter of sucrose solution and boil it with Benedict's solution. Okay, and confirm it that it doesn't give any, what is it? Any precipitate. First thing you have to do that one. So that is confirming sucrose is not giving any. Sucrose is not giving any precipitate. Thereafter, you need to do this. What do you have to do? Add 2 cubic centimeter sucrose solution to whom
1 cubic centimeter.
1 cubic centimeter whom 1 cubic centimeter whom
Dilute hydrochloric acid.
Dilute hydrochloric acid. It is helping to break the sucrose into whom monosaccharides. In exam paper, they will ask you, why do we add, okay, why do we add, we will put this one also to this, otherwise. So, what are you doing? Add 2 cubic centimeter of sucrose solution to 1 cubic centimeter of dill HCl aqueous and boil it for 1 minute. Boiling will make that breaking faster. What is the benefit of boiling? What is the benefit of boiling?
Helps in breaking the sucrose to monosaccharides faster.
Faster. Breaking of the sucrose will be faster when you boil it. Okay. Now what you have to do? Read the next one. Read the next one. Neutralize with whom? Neutralize with whom?
Sodium hydrochloride.
Sodium HCO3, sodium bicarbonate. NaHCO3 mean, who is it? Who is it? Sodium bicarbonate. What are you doing? Now HCl you have added, no. HCl you have added, no. Yes or no?
Yeah.
Yes, sir.
Now that medium is acidic medium. In acidic medium, Benedict's test will not answer. Benedict's solution will not give any precipitate if the medium is
Acid.
acidic. So after boiling, after breaking the sucrose into monosaccharides, we need to neutralize that. Sodium bicarbonate is a what kind of material? A base material. We are adding a base and neutralize it. After neutralizing, how do we know it is neutral? We are knowing that one with the use of whom? With
pH
the
paper.
use of pH paper. Thereafter, you carry out the Benedict solution. Observation. Now it has broken into monosaccharides, right? Now it has broken into monosaccharides, right?
Yes, sir.
Yes.
Yes.
Will it now give a brick red color precipitate mean?
Yes.
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 So a brick red color precipitate would be obtained. So previously it didn't give the precipitate, that we confirmed already. Now it is giving a precipitate that confirms it is a non-reducing sugar. Or you can call it is sucrose. Clear?
Clear, sir.
Please complete it down. Practical parts will come in the structure paper. Structure questions are giving you practical based questions. Completed? Have you completed, my children?
Yes, yes.
Right. Now we are going to work out the third one. The third one is test for home. Test for starch. Starch powder is in the laboratory, a white color powder. This experiment is a very simple experiment. Very simple experiment. You know this experiment from your small grades even. What is the experiment? What is the experiment? Tell me the procedure. What is the procedure? What is the procedure? You need to take 2 cubic centimeter of 1% starch solution in a test tube. As I told you, in the laboratory, you have starch powder. You need to take 1 gram powder, 1 gram powder, and dissolve it in 100 gram water. 1 gram powder should be dissolved in how much?
100 gram water.
Okay, so that is how we prepare that 1% starch solution. 1% starch solution. Okay, how do you make it? Actually, you have to do 1 gram starch dissolved in 99 gram starch or distilled water. But 99 gram is approximately how much?
100.
100 grams. So that's why 100 gram I'm choosing. So 1% starch solution, what is the meaning? What is the meaning? 1 gram starch dissolved in, what is it? 100 gram distilled water. And you are adding whom you are adding, few drops of whom you are adding?
ID.
Iodine. Iodine in your chemistry, inshallah, you will study what is this solution. This solution is the in normal words they call it by the name which solution? Iodine solution. Iodine solution is made with the mixing of I2 iodine solids and Ki potassium iodide. Okay, the combination of these two will be giving you, it is a brown color, reddish brown color solution. A reddish brown color solution. Okay, because it's experimental, no, Puta, you need to know their colors. You would have seen them. Okay, when you start your school life in advanced level, in schools, most probably school side they will do those practicals. Same time, I have all these materials with me. Okay, if you are interested, one day, inshallah, I will invite you all of you come, do the practicals, record the practicals, and keep with you. Clear?
Clear, sir.
Yes. So what is your observation? Okay, your observation is okay, dark blue or dark purple color solution will be obtained. Okay, actually, if your starch level is greater, that if you now 1% starch, no, 1 gram starch dissolved in 100-gram distilled water is a very dilute solution. Then only that dark blue color can be seen. If you increase the starch amount, that blue or purple will appear to you as black color. Actually, they are blue. Actually, they are purple. But how will they appear?
Black color.
Black. Okay. So that is the test for starch. Everything they will ask you. 1% starch solution mean what's the meaning? Okay, what are the chemicals present in the iodine solution? What is the color of the iodine solution? Okay, how many cubic centimeter you are taking? Everything is important. You, inshallah, do it practically. Okay, inshallah, we will arrange a time. Tell me after completing it. Completed?
Yes, sir.
Have
Yes, sir.
everyone completed it? Okay. Here, this is the practical. Remember, starch is a polysaccharide, Puta. Are polysaccharides water soluble? Are polysaccharides water soluble?
No.
No. So when you mix it, it will not easily dissolve.
Major nutrient.
It will make a milky material.
The main carbohydrates found in our food are in the form of starch and sugars. So we can easily test if a food item contains starch. For that we need iodine solution
And starch solution.
and a food item. It contains starch. Then we put 2 to 3 drops.
See the color of iodine solution. What is the color coming?
Brown color.
Brown color, reddish
iodine
brown.
solution into that food item. Then notice the
Can you see the color?
Yes, sir. Yes, sir.
Do you see the bluishness there?
Yes,
Yeah.
sir.
color change. So we can see
Enough.
the color becomes now a blue-black color. So it indicates that that food item contains starch.
That's all. Clear?
Clear, sir.
Clear. Now we are going to work on the next heading, that is test for lipids. Test for, what is it? Test for home lipids. Okay. To find out lipids, we have a simple experiment. It is called Sudan 3 test. What is the name of the test? What is the name of the test?
Sudan 3
Sudan
test.
3 test. Sudan 3 is a Sudan 3, if you consider. Sudan 3 is a powder. I have Sudan 3 with me. Okay. Sudan 3 is a powder. A water insoluble powder. What kind of powder? What kind of powder?
Water insoluble powder.
Water insoluble powder. This is that powder. What is the color of the powder?
Red color.
Red color. You need to make Sudan 3 solution. What do you want to make? What do you
Sudan
want to
3
make?
solution.
Sudan 3 solution. Can you dissolve Sudan 3 in water and make that solution?
Can't.
Can't.
No.
Why? Why? Because Sudan 3 is not
Polysaccharide.
water soluble. So what we do is we dissolve it in pure ethyl alcohol. In which material?
Ethyl alcohol.
Ethyl alcohol. That is how this solution is prepared. Okay, so after preparing that solution, you can go for the test. Sudan 3 test for the home lipids. This is a Sinhalese one. Okay, too long. I don't like lengthy videos because we don't need to spend too much time.
The Sudan 3 test, so we're testing for the presence of lipids in our McMush. So if you notice, I have a beaker of water. I'm going to take my Sudan 3, and when I put it into the water, let me move this around so you can see it. When I put it into the water, the Sudan 3 dissolves in the water. You can't really see it anymore. The water is turning slightly pink just because I put so much in it. But when I go to put this in some oil, so I'm going to take a couple pipettes of oil into my test tube. When I put the Sudan 3 into the oil, it stays on top and it will eventually slowly dissolve through. You can kind of see it starting to dissolve in there, but it stays pink. So we're going to see what happens when we do the same.
So it's a simple practical. When Sudan 3 get mixed with oil, the oil will be turning to a bright red color material. What is the experiment? What is the experiment? What are you adding to the oil?
Sudan 3.
Sudan 3 solution. The oil will be turning to
Bright red.
bright red color. That's all the experiment. But the real experiment is, this is a little longer video. Okay, wait a minute. Doesn't matter. You have time. Okay.
ක්ෂණයක් එක්ක. අද අපි කතා කරන්න යන්නේ ආහාරවල තවත් සංඝටකයක් වෙන තෙල් සාමයේද එහෙම නැත්නම් ලිපිඩ හඳුනගන්නේ කොහොමද කියන කාරණා.
He is showing you from the beginning how to are the Sudan 3 and how to do this experiment?
ඕනෙම සයින්ස් ප්‍රක්ටිකල් එකට කලින් පර්සනල් ප්‍රොටෙක්ටිව් ඉක්විප්මන්ට්ස් යූස් කරන එක හරිම වැදගත්. මොකද මේ සමහර කෙමිකල්ස් අපේ ශරීරයට ගොඩක් අහිතකර වෙන්නත් පුළුවන්. මුලින්ම අපි සුඩන් 3 සොලියුෂන් එක හදාගමු. සොලියුෂන් එක හදාගන්න සුඩන් 3 පවුඩර් එකකින් ස්වල්පයක් එතනෝල් වල දිය කරගන්නවා. දැන් මේ මිශ්‍රණය අපි ෆිල්ටර් පේපර් එකක් ආධාරයෙන් ෆිල්ටර් කරගන්න ඕනි. මේ ක්‍රියාවලිය හොඳට වාතාශ්‍රය ලැබෙන තැනක කරන්න අමතක කරන්න එපා.
So this is how we prepare the, what is it?
Sudan 3 solution.
Sudan 3 solution. We are dissolving Sudan 3 powder in ethanol, ethyl alcohol.
සියලු දේවල් අපි පහළ ඩිස්ක්‍රිප්ෂන් බොක්ස් එකේ දාලා තියෙනවා. දැන් අපි ටෙස්ට් කරන්න අවශ්‍ය සාම්පල් ටික හදාගමු. මුලින්ම අපි චිකන් සාම්පල් එක හදාගන්න අපි මේ අරන් තියෙන චිකන් පීසස් ටික පිස්ටල් ඇන්ඩ් මෝටර් එක ආධාරයෙන් ග්‍රයින්ඩ් කරගමු.
He is testing for any food item having lipid. We'll see.
මේ සාම්පල් ටික ද්‍රව විදියට තියෙන එක අපිට පහසුයි. පිස්ටල් ඇන්ඩ් මෝටර් එකෙන් චිකන් ටික ග්‍රයින්ඩ් කරන එක අපි හිතපු තරම් ලේසි වුණේ නැහැ. සෑහෙන වෙලාවකට පස්සේ චිකන් ටික ග්‍රයින්ඩ් කරගන්න කොහොම හරි අපිට පුළුවන් වුණා. අපි මේ දේ පරීක්ෂා කරන්න බටර් සාම්පලයක් පාවිච්චි කරන්න හිතුවා.
Adding some lipid butter.
මීළඟට අපි බටර් සාම්පල් එක හදාගන්න බටර් ටික බන්සන් බර්න් එක ආධාරයෙන් මෙල්ට් කරගමු. ඉතින් සයින්ස් වලට කැමති ඔයාලා වෙනුවෙන්ම අපි හදලා තියෙන Telegram group එකට එකතු වෙන්න ඔයාලටත් පුළුවන්. Telegram group එකට join වෙන්න අවශ්‍ය link එක පහළ description box එකේ දාලා තියෙනවා. දැන් අපි samples තුන test tube වලට දාගමු. මුලින්ම butter sample එකත් ඊළඟට chicken sample එකත් අවසාන test tube එකට මුලින්ම ආශ්‍රිත ජලයත් ඊළඟට තෙලුත් එකතු කරනවා. මේ වගේ තවත් විද්‍යා පරීක්ෂණ බලන්න ඔයාලා කැමති නම් The Academy SL subscribe කරන්න අමතක කරන්න එපා.
Coconut oil. Sudan 3.
මීළඟ පියවර විදිහට Sudan 3 solution එක අපි හදාගත්ත samples තුනට එකතු කරනවා. කිරලබ වලින් ටෙස්ට් ටියුබ්ස් ටික හොඳට කවර් කරලා සොලියුෂන්ස් ටික මික්ස් කරනවා. සොලියුෂන්ස් ටික මික්ස් කරාට පස්සේ අපිට නිරීක්ෂණ වලට එළඹෙන්න පුළුවන්.
Can you see the observation? Can you see the observation?
මේ තෙල් සහ ජලය
Yes,
නම්
sir.
In the first test tube, at the top, there is oil. Below that one, there is water. Does the water get the Sudan 3?
පරීක්ෂණාලේ තෙල් ඇති කොටස පමණ සුඩන් 3 මගින්
Does the water get the Sudan 3?
No.
No, it is staying as a transparent material. The only oil part is collecting the Sudan 3. Correct?
ආශු ජලය තෙල් සහ මේද අඩංගු නැති
Yes.
නිසා ජලය ඇති කොටස සුඩන් 3 මගින් වර්ණ ගැන්වෙන්නේ නැහැ. බටවල මේද අඩංගුවන නිසා මේ බට ද්‍රාවණය සුඩන් 3 මගින් රතු පැහැයෙන් වර්ණ ගැන්වී තිබෙනවා. චිකන් සාම්පල් එකේ තෙල් සහ මේද අඩංගු නිසා චිකන් සාම්පල් එකත් සුඩන් 3 මගින් වර්ණ ගැන්වී තිබෙනවා.
So this is your observation. Clear?
Yes, sir.
Right. So we have writing wise, it is a short one. Writing wise, it is a short one. Okay, understand, have to understand it, no? Okay, that's very important. That is very, very important. Okay. So what is the experiment? See, add 2 cubic centimeter of oil to 2 cubic centimeter of water in a test tube. Here oil will be staying where? Above or below?
About.
Above. Add few drops of Sudan 3 and shake. This is the procedure. This is the procedure. Okay. If this is the procedure, what is your observation? The oil layer that stays on water would turn to what color? Bright red color. Correct?
Correct, sir.
Completed.
Done, sir.
Good. Now coming for the next experiment, that is test for proteins. Test for whom? Test for
Protein.
proteins. Test for proteins is also called by the name biuret test. What is the name?
Biuret test.
Biuret test. So first of all, we will go to see the biuret test for proteins. Thereafter, we will come back to the procedure. Yeah, biuret test for proteins.
අපි හැමෝම දන්න විදිහට ප්‍රෝටීන් කියලා කියන්නේ ආහාරවල තියෙන ප්‍රධාන සංඝටකයක්. ඉතින් ප්‍රෝටීන් හඳුනගන්නත් ඒටම අදාළ විශේෂ පරීක්ෂණ කීපයක් තියෙනවා. ඉතින් අද අපි මේ කතා කරන්න යන්නේ මේ පරීක්ෂණ ගැන. ප්‍රෝටීන් හඳුනගන්න ප්‍රධාන වශයෙන් පරීක්ෂණ හතරක් තියෙනවා. මේ පරීක්ෂණ හතර මම කිව්වොත් පළවෙනි එක බයුරේට් පරීක්ෂාව, දෙවෙනි එක මිලන් පරීක්ෂාව, තුන්වෙනි එක සැන්තෝප්‍රෝටීක් පරීක්ෂාව සහ ඊට අමතරව අපි ගාව තියෙනවා නින්හයිම් රීන් පරීක්ෂාව. ඉතින් මෙන්න මේ පරීක්ෂණ වර්ග හතරෙන් ප්‍රධාන වශයෙන් භාවිත වෙන පරීක්ෂණය වෙන්නේ බයුරේට් පරීක්ෂාව. ඉතින් අද අපි මේ වීඩියෝ එකේදී කරලා බලන්න යන්නේ කොහොමද ආහාර ද්‍රව්‍ය වලට බයුරේට් පරීක්ෂාව සිදු කරන්න කියන කාරණය ගැන. ඉතින් මෙන්න මේ ප්‍රෝටීන් පරීක්ෂාව සිදු කරන්න අපි පාවිච්චි කරන බයුරේට් ප්‍රතිකාරකයේදී මේ බයුරේට් ප්‍රතිකාරකය හදාගන්න අපි පාවිච්චි කරනවා සෝඩියම් හයිඩ්‍රොක්සයිඩ් සහ කොපර් සල්ෆේට් ද්‍රාවණය. ඉතින් මෙන්න මේ දෙන්න ප්‍රෝටීන් එකක් ප්‍රතික්‍රියා කරාට පස්සේ මේ කොපර් සල්ෆේට් වල තියෙන ලා නිල් පාට දම් පාටක් බවට හැරෙනවා. ඉතින් මෙන්න මේ කාරණේ තුලින් අපිට හඳුනා ගන්න පුළුවන් ඒ අදාළ ආහාරය තුල ප්‍රෝටීන් තියනවද නැද්ද කියන කාරණය ගැන.
It is egg white. What is it?
Egg white.
Egg white. That is sodium hydroxide. So first you are adding
Sodium hydroxide.
now copper sulfate solution, few drops. What is the color coming?
Purple color.
Purple color. And in the previous experiment, there is test for whom? Test for lipids. Have a look on the rest.
ඉතින් අපි අවසානයේදී මේ චිකන් සාම්පල් එක අන්වීක්ෂයක් ආධාරයෙන් පරීක්ෂා කරලා බලමු. දැන් අපි චිකන් සාම්පල් එක අන්වීක්ෂය ආධාරයෙන් නිරීක්ෂණය කරමු. ඩ්‍රොප් එක ආධාරයෙන් චිකන් සොලියුෂන් එකකින් සුළු ප්‍රමාණයක් අරන් අන්වීක්ෂ කදාවක් මත තියලා වැසුම් පැත්තකින් කව කරලා අන්වීක්ෂ වේදිකාව මත තියලා නිරීක්ෂණය කරනවා.
observing it under microscope. He has a nice camera called Pro 7. He is the one told me. The Pro 7 camera, you can keep that camera on the, what is it? Microscope and take a photograph. I spoke with him, then he told me that. So, using that one, he is taking a photograph. He's taking a photograph. This is how that photograph looks like. Can you see the pink color? Circular component?
Yes, sir.
They are the lipid droplets. They are the, what is it?
Lipid droplets.
Lipid droplets. So previous to the protein heading, when these Sudan, when the Sudan 3 lipid plus water sample is observed under microscope, right? Pink color, spherical droplets can be, what is it? Noticed. Clear?
Clear, sir.
Good. Complete it.
ரண்ணா வெளியே காய் போடுவானா உள்ள கம்மியில போட சொன்னேன் ஏன்னா அவங்க கேமரால எல்லாம் பாத்து பண்ணிப்பாங்க
Completed the writing?
Yes, sir.
Right. So, now let's try it for the protein test. What are you doing? What are you doing? Add 2 cubic centimeter protein solution to volume, equal volume of 5% potassium hydroxide. 5% potassium hydroxide means take 5 gram potassium hydroxide, dissolve in 100 gram water. Remember, now you have to do that one proportionally. Now you need little potassium hydroxide, correct? 2 cubic centimeter, 2 milliliter. 2 milliliter means 2 gram like. In that case, do you want to prepare 100 gram solution?
No.
No. 5 gram dissolved in 100 gram mean 0.5 gram dissolved in 10 gram. Correct?
Correct, sir.
So if you use 0.5 gram, measure and take 0.5 gram dissolved in 10 gram distilled water, Preparation same, 5%. Clear?
Clear, sir.
Similarly, copper sulfate solution, you need only 1%, 1 gram copper sulfate dissolved in 100 gram. Then 10 gram mean 0.1 gram. Little only you need. No need to waste. Chemicals are very expensive. We have to very carefully handle them, and we have to economically handle them. Okay. Now, procedure is ready. What is your observation? Okay. A purple color would be observed. Understood? A purple color would be, what is it?
Observe.
Observed. Understood? Complete it,
Yes, sir.
complete it.
Done, sir.
Completed.
Yes, sir.
Good. So that is the experiment for home proteins. Correct? And finally, a note for you. A notice. The note is also a practical part. What is the heading? What is the heading? What is the heading?
Preparation of iodine solution.
Preparation of iodine solution. Take 71 gram iodine crystals. 71 gram, bigger number. Understood? 10 gram, 3000. Okay, expensive. So we have to do it proportionally, as I told you. Okay, 2 gram potassium iodide and dissolve them in how many cubic centimeter?
300 cubic
300 cubic. And now if you are going to 30 cubic centimeter, each one will be divided by 10. 0.2 gram potassium iodide, 7.1 gram iodine crystal, like that. Clear?
Clear, sir.
Hmm. Okay, completed.
Yes, sir.
So with this one, the practical portions are over. Related to whom? Related to whom? Chemical basis of life. Okay. Now we are going to today start which portion? Which portion?
Cellular basis of life.
Cellular basis of life. So if you go for cellular basis of life, it would have certain experiments, but most of those experiments are visual experiment, that means pictures, okay, like experiment, not these kind of laboratory experiments are there in the cellular basis of life, right? So take the cellular basis of life, tute. Okay. Remember, I have sent that message also to you. Anyway, that is my responsibility to again emphasize you. Please take the hard copy printers, printed materials and maintain the materials. Some of you have, what is it, pads, iPad like things. I can use this one. So if don't mistaken me, if the iPad falls and breaks, everything is over, right? Do we have any surety it will never happen so? Tell me.
No, sir.
No. So point it is, always maintain hard copies. If I'm sending essays to you, essay schemes I'm sending, take printout of those schemes, maintain a file. These are the essays. So take a printout and paste that one with your essay. You have written an essay, you have corrected the essay. With that corrected essay, my scheme. Clear? With that corrected essay, my scheme. You are maintaining. Now, in the beginning, I told you, alone in chemical basis of life, there are more than 6 essays to complete. Okay, more than 6 minimum. Okay, so we are here going to speak about the cellular basis, that means cells components, okay, what they do, what is cell, okay, like cell related life components we are going to discuss in this chapter. Okay, so what is the heading for you? Beginning heading, historical background of the cell. We will go for that. You know this picture, correct? In your O-level grade 10 book had this picture. The man who first discovered cell and called it cell using his own microscope was Robert Hooke. The cells he noticed were dead cells. Actually, what he observed under his microscope, a
piece of coke.
A piece of coke. The cells present in a coke are what kind of cells?
Dead cells.
Dead cells. So the first observed cells are
Dead cells.
dead cells. When he observed it, as in this right side picture, he noticed several cage-like structures. Then he concluded, living organisms are made of the basic units called, what is it? Cell. Cells in Latin meaning, boxes, cages like meaning. Okay. Nowadays, even in prison, the rooms where the prisoners are living are called by the name, what is it? Cell. Okay. Anyway, this can be a small support to an MCQ. This could be a little support to a small support to an MCQ or structure question. A little, very little. 1 out of 10,000 contribution there for this knowledge in the exam paper. But this knowledge has a contribution in the exam paper. Second person who is contributing to cell biology is whom? Read his name.
And
Read his Anton van Leeuwenhoek. This man, he prepared his own microscope and observed pond water. What he observed? He observed pond water. When he observed pond water, in that pond water, he observed microscopic organisms like euglena and bacteria. He gets the next honor, observed microorganisms for the, what is it? First time. Due to this reason, he is described by the word father of microbiology. Okay. He is described as the father of, what is it?
Microbiology.
Microbiology. Clear?
Clear, sir.
Could you please mark them down? Marked it?
Yeah, sir.
Yes. Now, the well-known three names. You are familiar, right? The three people who contributed to cell theory. Who are the three? First person, Matthias Scleden. Second person,
Theodore Squan.
Theodor Schwann. Third person, Rudolf Virchow. The first concept of cell theory came in the mind of Matthias Cleiden. The other people really joined him. The concept of cell theory first came in the mind of Matthias Cleiden. Matthias Cleiden was basically whom? Basically a botanist. So botanist means the person who studies
Plants.
plants. So his study area is on plants, not animals. So his personal conclusion is this. His personal conclusion is this. What is the personal conclusion of Matthias Cleiden?
All plants are made up of
All
cells.
plants are made up of cells.
Cells.
So in exam paper, if they ask you what is the opinion of Matthias Cleiden, what is your answer?
All plants are made up of
All
cells.
plants are made up of cells. Theodor Schwann, he was a zoologist. He studied the animal area and did this conclusion. What is the conclusion?
Animal tissue.
Ah,
Also made up of
animal tissues are also made up of cells.
Cells.
Now see, the delivery of Matthias Cleiden and Theodor Schwann are not looking alike. Theodor Schwann says animal tissues are made up of cells. Matthias Cleiden says all plants are made up of cells. Correct? Right.
Yes, sir.
And Virchow, he made this decision individually. What is the decision?
All cells rise from
All cells arise from pre-existing cells by cell division. Okay. Now these three people came together. They collaborated. They joined hands together and gave us the cell theory. What are the three portions of the cell theory?
All organisms are composed of one or more cells. The
All
basic structure and
organisms
function.
are, whether it's plants, whether it is animals, whether it is microorganisms, all organisms are composed of one or several cells. Second one,
The basic structural and functional unit of organisms is the cell.
the basic structural and functional unit of organisms is the cell. Third one,
All cells arise from pre-existing cells.
pre-existing cells. Clear?
Clear,
Right.
sir.
Remember, in any cell, if you take any cell, plant cell, animal cell, microorganisms cell, mean there will be 5 major components. How many components? How many
5.
components? 5 major components. One component's name is
Plasma membrane.
plasma membrane. Second component's name is
Cytosol.
cytosol. You can call it cytoplasm also. They are using the word cytosol. Third component's name is
Subcellular components.
any material staying inside the cytosol are coming under, or any
Subcellular.
material within the plasma membrane is coming under subcellular component. Next one,
DNA.
DNA. Actually, DNA is a subcellular component, but specially mentioned because it's a genetic material. Okay. And next one,
Ribosomes.
it is also a subcellular component. very important for a cell survival to synthesize proteins. So basically, what are the components making the cell, any cell? Any cell. What are the components? Tell me. Number one,
Plasma membrane,
plasma membrane,
cytosol,
cytosol,
subcellular components,
subcellular component,
DNA,
DNA,
ribosomes.
ribosomes. Okay. A small description is given to you regarding plasma membrane. A small description is given to you regarding cytosol. Okay. A small description is given to you regarding DNA, and ribosome, they have nothing special given. But remember, DNA, ribosome, both are what kind of components? Subcellular components. But they are very special. DNA and ribosome are subcellular components, but very, very, right? Now, it's like this. Okay? It's like a family member. Everyone is, there are so many people we can call family member, correct? Am I right?
Yes, sir.
But Dada and Mama are special, correct?
Yes, sir.
They are also family members. But just you can't tell, Dada is a family member. But can you just tell who is he? He's a family member?
No.
No, no. He is very special for us. Mama is very special for us. Is it like that? DNA, ribosome, both of them are, what is it? Subcellular components, but they are very special. That's why they have separately mentioned them. Otherwise, they also will come under subcellular component. Okay. So please mark it down. Have you marked it?
Yes, sir.
Yes. So what is the description given to you regarding plasma membrane? Two descriptions are given to you. One, it is telling plasma membrane is bounding a cell. So the external limit of a cell is home.
Plasma membrane.
Plasma membrane. Another description given to you, it is what kind of barrier? A selective barrier. Why should we call it selective barrier? It will allow materials to go into the cell and to go out of the cell selectively. Who should go in? That one can go in. How much should go in? That much can go in. Who should go out? That one can go out. How much should go out? That much should go out. Control. Clear? Anyone can go, any much can go mean, is it selective barrier?
No.
No. Like your advanced level. Can anyone go to the medical faculty?
No.
Can everyone go to the medical faculty?
No.
No. Okay, these people in this number, if you take Kandy district, nearly 100 students are collected every year to the medical faculty. Nearly 100. Okay, nearly 100 are picked for the medical faculty. I'm not going to speak more detail. So, who is selected? The talented ones are selected. How many selected? Only 100 are selected. So, your university entrance is a selective barrier. Understood?
Yes, sir.
Yes. Second one, cytosol. What are the descriptions given to you? Cytosol. It is a semi-solid, semi-fluid, semi-solid or semi-fluid, both are same. Jelly-like, both are same. Semi-fluid, if you say, no need to say jelly-like. Jelly is semi-fluid material. Understood? Semi-fluid
Yes.
jelly-like material. It is occupying the area within the plasma, okay, covered by the plasma membrane. Plasma membrane is actually covering the cytosol. Now, take like this. At your home, do you have a fence or a fence wall? Do you have a fence wall?
Yes.
Yes. Within that fence wall, there is a land, correct?
Yeah.
Yeah. That land, in that land, one place is occupied by your house. Another place will be occupied by a tree. Okay. External things will be there, internal things will be there. Yes or no?
Yes, sir.
So, all these together are outside surrounded by the fence wall. Like that, the land is like cytosol. The fence is like plasma membrane. Clear?
Clear, sir.
Third
Clear,
one,
sir.
what kind of components?
Subcellular components.
Sub mean within. Okay, components within the cell. Any component, anything present, fixed to the, placed inside the cytosol. It is called
Subcellular components.
subcellular component. In your O level, you studied mitochondria, chloroplast, Golgi apparatus, endoplasmic reticulum, ribosome, lysosome, like several things you studied. Here you will fully detail study that. DNA. DNA's description is
Genetic materials.
genetic material. Ribosome's description is, it is protein synthesizing subcellular component. What is it? What is it?
Protein synthesizing.
Protein
subcellular component.
synthesizing subcellular component. Clear?
Clear, sir.
Okay, it's already a protein synthesizing machine. Protein synthesizing machine is called
Ribosomes.
ribosome. If proteins are not synthesized, a cell cannot perform metabolism. If metabolism cannot be performed, can it be a living cell?
No.
No. Are you clear?
Yes, sir.
Yes. Passing that portion, we are coming to an important site. In this place, we will be learning lots of details. In the cells, there are two types of cellular organizations. What are the two types of organizations? One is called
Prokaryotic.
prokaryotic organization. Next one is called eukaryotic organization. If you take these two organizations, okay, look at the two pictures there. Just see the pictures. Left side picture is a prokaryotic organization. What are the components present in a prokaryotic organization? Mean there is a cytoplasm, correct? Can you
Yes,
see
sir.
cytoplasm? There is a region called nucleoid. Can you see?
Yes, sir.
Yes. There can be flagella, long extensions. There can be pili, short extensions. There will be a plasma membrane. They have called here cytoplasmic membrane. That kind of a word is not in our syllabus. Okay, that is plasma membrane. Externally, in the prokaryotic cells, what do you notice? That green
Cell
color
wall.
part. What is it?
Cell wall.
Cell wall. This kind of a unit. Just have a look. Few words. Cytoplasm, nucleoid. Just read it. Cytoplasm, nucleoid, flagella, pili, ribosome, plasma membrane, cell wall like things. Here what do you have? What do you have? Yes. Just read it.
Ribosomes, nucleus, nucleolus, mitochondria, cytoskeleton, Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, plasma membrane, peroxisome, centrioles.
So who has more components? Eukaryotic or prokaryotic?
Eukaryotic.
Eukaryotic. Who is looking more complex?
Eukaryotic.
Eukaryotic. Who is looking large?
Eukaryotic, sir.
Eukaryotic. Right? Like this, there are two organizations. Between these two organizations, there are certain differences they are listing here. First, it is the organisms belonging to prokaryotic organization. Who are the organisms? Familiar words. They are not unfamiliar words. Familiar words they are. What are the organisms?
Bacteria.
Bacteria. Second one,
Archaebacteria.
archaebacteria. Okay. Eukaryotic one. What are the organisms? Protista. Protista. Fungi.
Plants.
Plants.
Animals.
Animals. Okay. Second one, prokaryotic cell size. Prokaryotic. What is it? Cell
Cells.
size. Have I written a size to you? Have I written a size to you? Average diameter for prokaryotes, is it written? Is it written?
Yes, sir.
Written here? Are you written the diameter?
No, sir.
No. Actually, in 2024 batch from the resource book, there's a correction. Okay. But unfortunately, English medium resource book in printable form has not yet been released. That is a problem. Okay, 0.5 to 5 micrometer. Can you read the eukaryotic size, my children? How do you read this part? How do you read this part?
10 micrometer to 100 micrometer.
Like that, left side prokaryotic size is 0.5 to 5 micrometer. Resource book has it as 1 to 5 micrometer. That has been now corrected. Clear?
Yes, sir.
Now, last time I told you one. Magnesium is considered under which elements? Trace elements. One of you asked, sir, magnesium is a macro element. No, that has gone now. Understood? Understood?
Yes, sir.
If you take in general, magnesium is what kind of element? What kind of element?
Trace element.
Trace element. Like that, there are corrections. Okay, the old resource book, some of you, okay, would have asked the seniors, anyone who spoke with seniors regarding the biology lesson or A-level science? Anyone spoke with you, any one of your seniors? Seniors mean the girls who are studying in the previous batches, your school students or anyone, your relative, anyone?
Yeah, sir.
Yeah. Did they tell you about the resource book?
Yes, sir.
Yes. Did they tell you how to follow only the resource book?
Yeah.
So thing is, you can't follow only the resource book. That is, we teachers decide it. Understood? According to the instruction coming from the government, we decide what is relevant and what is irrelevant. Okay, that resource book that your seniors are using up to 2023 batch is using cannot be used for 100% for you. So don't buy now. Don't try to buy any resource book now. Okay, what I am giving is much more enough for you. Much more enough for you. Okay. Can you understand the cell size? What is the cell size prokaryote? Prokaryotic cell size?
0.1 micrometer.
0. Tell me correctly. 0.5 to 5 micrometer. Have you marked that value? Have you marked that value? 0.5 micrometer. Micro I can't write. I write like this. Okay. Micrometer to 5 micrometer. Micro sign you know, correct?
Yes, sir.
Third one, form. Mainly prokaryotes are in what form? In what form? That means some of you whom? Few?
Multicellular.
Multicellular. Majorly unicellular, little, what is it?
Multicellular.
Multicellular. Here other side. Mainly whom?
Multicellular.
Few?
Unicellular.
Unicellular. Who are the organisms coming under unicellular forms of eukaryote? Most of the protista. You studied in your O-level amoeba, paramecium, euglena, chlamydomonas, like protista, and some fungi like yeast. Some fungi like, what is it? Some fungi like
Yeast.
yeast. We will later take that one. Have you marked this red color portion? Few are, what is it?
Multicellular.
Multicellular. Third, it is evolutionary origin on this earth. The first organism evolved was a prokaryote. How many years ago it evolved?
3.5
3.5 billion years ago it evolved. Eukaryotes evolved how many years ago?
1.8 billion years ago.
That means from 3.5 billion years to 1.8 billion years, for 1.7 billion years on this earth, who was there?
Prokaryotes.
Prokaryotes. Only prokaryotes were living on this earth for how many billion years? How many
1.7.
billion years? 1.7 billion years. Clear?
Clear, sir.
Yes, sir.
Good. So, third it is their cell division pattern. In the prokaryotes, you know, there's no nucleus. There's no, what is it? In the prokaryotic cells, there is no, what is it?
Nucleus.
Nucleus. Do you know these words? Do you know these words?
Yes, sir.
Yes. These words are in your O level cell divisions, right? Yes
sir.
or no? In your A level, these ones are actually whom?
Nuclear division.
Nuclear divisions. Mitosis is a nuclear division, nucleus dividing. Meiosis is also a, what is it? Nuclear division. Do we notice nucleus in prokaryotic cells?
No.
No. If there is no, okay, nucleus in the prokaryotic cell, can you expect nuclear division there?
No.
No. Binary fission. Binary fission, it is a cell division. Who is dividing? Who is dividing?
Cell.
Cell, not nucleus. But here, okay, cell division, if you consider, it is equal to nuclear division plus cytoplasmic division. These two divisions together only we call here cell division. But their cell division mean only cytoplasmic division will be there. Understood?
Yes, sir.
Understood the idea? Is it clear for everyone?
Clear, sir.
Clear. Could you please mark these details then? Completed?
Yes, sir.
Yes. Right? For an idea, I will show you the binary fission in bacteria. Okay. Binary fission in bacteria. I will show you a small photograph for you to take a clear idea. Okay. Because binary fission also a familiar word to you from your grade 10, but you don't know really what is binary fission mean. They told binary fission, you took it as what's it? Binary fission. That's all happened. But here that is not the right thing. For every word, we have to ask that question, what, why? Right. So overall, let's have a look on the bacteria's binary fission. Okay. One bacteria, when it undergoes binary fission, first of all, its DNA, it has a DNA, correct? Bacteria, no? Bacteria has, what is it?
DNA.
DNA, its DNA is replicating, 2 copies are made. Now, cell divides, cytoplasm divides. DNA replication, then cytoplasmic division. Finally, how many cells of bacteria coming? How many cells of bacteria coming? 2 cells of bacteria coming. Do you see a nucleus here?
No.
No. Nucleus is not there, just the DNA there. That DNA undergoes, what is it? A replication. So nucleus means a very complex structure. Nuclear division means a very complex process. Bacteria is a simple organism. So simple organisms performs in what manner? Simple manner. Clear?
Yes, sir.
Right. So that is the idea behind cell division. Thereafter, genetic material. If you take the prokaryotic genetic material, it is described with this word, my children. What is the word?
Circular.
Last class, I showed you a circular DNA, right?
Yes, sir.
Yes. This DNA, circular DNA, is located in which place?
Cytoplasm.
Cytoplasm. The region where the DNA is noticed is called by the name.
Nucleoid.
Is it covered by or is it associated with proteins?
No. No.
No. To that reason, how do you call the DNA?
Naked.
Naked DNA. Okay. If you go for the eukaryotes, circular opposite word. What is it?
Linear.
Linear DNA. Where it is located?
Nucleus.
Nucleus, not in the
Cytoplasm.
cytoplasm. It is associated with
Proteins.
proteins. Here, an important point you have to write. Okay. Circular naked DNA are found in mitochondria and chloroplast of whom?
Eukaryotic cell.
Eukaryotic cells. Eukaryotic cells also have circular DNA, but not in the cytoplasm, not in the nucleus. Where can you see that one? Inside the mitochondria, inside the chloroplast. Mark that point there. Very useful point for you in exam papers, MCQ points. Okay.
Okay, sir.
Good. So in that place, we are finishing our session today. Inshallah, we'll meet. The works will come to you. Do the works. Okay. Keep the spirit and continue the works. Bye. Assalamu alaikum.